Hey, what's up guys? This is Adi from Shastri Nation, and today we're going to learn about cellular respiration. So cellular respiration is how our cells digest the things we eat and make them into ATP, or usable energy. So this happens in both plant cells and animal cells. So in plant cells, this happens in the mitochondria as well as in animal cells right here. Mitochondria. Alright, so this happens in the mitochondria and the cytoplasm. The cytoplasm is where the first step happens, or glycolysis. We'll learn about that more in the next couple of minutes. And the mitochondria is where the rest of it happens. So the mitochondria will happen in the inner matrix and somewhat in the outside here. Okay. Okay, so let's talk about the 3 stages of cellular respiration. So there are 3 main stages. There's glycolysis, which is the first stage, and in this this is going to happen in the cytoplasm, right here. And then we have stage 2, which has 2 names, which is the Krebs Cycle or the citric acid cycle. The citric acid cycle is because that's what it makes, and then the Krebs Cycle is named after the guy who found it. Then we have the electron transport chain. This and step 3 and step 2, or stage 3 and stage 2, both happen in the mitochondria. Now one of them happens in the inside and the other one happens on the middle. Alright, so let's talk about glycolysis. Alright, glycolysis is really a 9-step process in which we go from glucose, use 2 ATPs, and then we gain 2 ATPs and we make this thing called pyruvate. Now this pyruvate is going to go on to the next section, the next step, and then you'll see what happens there. But really the main things that we've made are the pyruvates, the 2 ATPs, and the 2 NADHs. So let's talk about the 2 products. The 2 NADHs. Now NADH is just an electron carrier, so electron. So what it basically does is it just carries electrons to step 3, or the electron transport chain. And you have a net gain of 2 ATPs. So really you make 4 ATPs. You make 4 ATPs, but you use 2 in the first place, so you end up with 2. That's why we have a net gain. Okay, and then we have 2 pyruvates, which is basically just glucose cut in half. So this would be a 3-carbon molecule. Well, glucose is a 6-carbon molecule. Okay, so then we have the link reaction, or the Krebs cycle. Okay, so the link reaction that leads to the Krebs cycle, my bad. So the link reaction, what happens is that you have you start out with the pyruvate, and then you have NAD being oxidized to form, reduced to form NADH. I always get confused with those. Anyways, you have those reduced to form NADH plus H, and then you have CoA, this enzyme called acetyl-CoA, coenzyme A, comes in and bonds to this pyruvate and releases CO2. So that forms acetyl-CoA, which will then go into the actual Krebs cycle, which is very, very, very complicated. All you need to know is that a lot of NADH is made, like here, here, and here, and FADH is made right here, and then some ATP. Some ATP. Alright, so what happens is that you have this thing where you add carbons in, and then you take them out to make the NADH, FADHs, and ATP and stuff like that. And that's why it's called the Krebs cycle, or the citric acid cycle, and because it happens so in a cycle, as you see here. It happens in a circle. Alright, so then let's talk about the products now. So you have 8 NADHs. So again, these are all going to come in handy when we go to the electron transport chain. So you have 8 NADHs, you have 6 CO2s. Now this is what we breathe out, and so what will happen is this will go out the cell and then go into our bloodstream and then go back to our lungs and go out from there, basically. And then you have 2 ATPs. So I mean, not much, but for right now, how many ATPs we've made is we've made the 2 ATPs here, and then we've made the 2 ATPs from glycolysis. So that totals to 4 ATPs. Not a lot, but you'll see in the last step how many we actually make. Now, FADH2s are also electron carriers. They just produce less energy. You'll see why that happens. Alright, so let's talk about the electron transport chain. Now this is the mitochondrial intermembrane space. So this would be the inside, if we drew a mitochondria. Well, there's my poor depiction of a mitochondria. And this would be the mitochondrial matrix, and this would be the inside. This would be the middle, but not like the outside, Alright. So then what we have to do is, you have, like right here, you have the NADH. The NADH comes in through here. Oh, sorry. NADH comes in through here, and then it drops off a full hydrogen atom and with the 2 electrons, so it's not just a proton. And that atom goes into the intermembrane space. So you have a H2 minus, sorry. 2 minus. Now you have these little proteins that only accept electrons. So the electrons, the hydrogen will stay back here. The electrons will go through the enzyme and come back on the outside of the membrane. Then the membrane will then those electrons will then bond again to a hydrogen, making it 2H2 minus. Sorry, I don't know what I'm doing there. Yeah. H2 minus, and then that atom will go through here, and you get the idea. It's same thing happens. This enzyme too only accepts electrons, so then you have electrons coming out here again. And then you have the last step. Oh, sorry. You have the electrons coming out here, and then you have the last step where we go to the inside here, and then we have we have the hydrogen atom bond to the to 2H2O, or to make 2H2O, sorry. So you have a half O2, and then you have a hydrogen come in, and that makes H2O. Sorry, 2H2O. A 2H2O. Yeah, sorry about that. Anyways. So that's what that happens. And now, if you haven't already noticed, there's a high concentration of hydrogen ions here. And so, since there's a concentration gradient going this way, the ions have to diffuse across this way in order to maintain equilibrium, right? So, they can't just diffuse across the membrane because they won't go through. It's not possible. So what they have to do, they have to use this thing called ATP synthase. And as you can probably see, ATP synthase makes ATP synthase. It's pretty, yeah. So ATP synthase makes energy so these H ions will go through this, and you have a little turbine thing that spins, and you have a ADP, as you see here, an ADP getting slapped on to a phosphate group, and that makes the ATP ion. Alright, so basically what happens is, you have the electrons, you have hydrogen, you have a high, to recap, we have a high concentration of hydrogen ions, and those will go through ATP synthase, like this, and then make ATP. And so this will be, the ATP will end up in the outer inside thing. I don't know. I mean, like, it won't be in the matrix of the cell, it'll be in the outside middle thingy. Yeah, I guess. Yeah. Okay, so now what, we talked about NADH, but we haven't talked about FADH, right? Alright, so why FADH makes less energy, I guess, in a way, is you know how NADH went through the first protein molecule, carrier protein. FADH starts with the second one, right here. So this, actually not that one. I don't know what that is. Yeah, this one goes through here, or like later in the process, and that's why less hydrogen ions are put on the intermembrane mitochondrial space, thereby making the production of ATP lower because of that. Yeah, I mean, I guess, okay, so like, the more steps you go through, the more hydrogen ions you put on the intermembrane space. And so, if you start out in later steps, the less you do, because then you would have to bond with oxygen and make water. So that's what I'm trying to get at, I guess, if that makes any sense. If it doesn't make any sense, just ask me in the comments below. That'd be great. Alright, so let's go ahead and talk about the products. So the products are about 36 ATP. It's debatable because there's a lot of debate about it because of the factors that are involved. And some say it's about 32, but I think it's about like 36. So okay, now I've only you might be asking, hey, you've only talked about glucose. What about other things? We eat other things, right? We eat proteins, fats, and other polymers and biological molecules, biomolecules, And yeah, I'll answer your question on that. So, but basically, what happens is you have the two molecules. You have fats. You have fats, and then you have proteins. We're going to talk about fats first. So what happens with fats? Fats are either converted into PGAL. Yeah, okay. I don't know why it's writing. Okay. PGAL or acetyl-CoA. So what is PGAL and acetyl-CoA? So, I mean, if you remember, acetyl-CoA goes back to the link reaction, and then that's what is going to put it into the Krebs cycle. So that's where it's going to continue off. It's going to continue off in the Krebs cycle. Oh. It's going to continue off in the Krebs cycle. Now PGAL, on the other hand, will continue in the first stage, or glycolysis. PGAL was made in the glycolysis. Phosphoglyceraldehyde. So basically what I'm trying to get at is these fats and proteins will be converted into other things and then just continued along the same process. So that's why we talked about glucose, because it's the basis of everything. Alright, so now let's talk about proteins. Sorry, I got ahead of myself there. But let's talk about proteins. Now proteins, again, are converted into pyruvates. If you remember, that's from glycolysis. And then or acetyl-CoA. So this will go back into the Krebs cycle. This will go into glycolysis. These two will go into glycolysis. This one will actually go into the link reaction, but, it's from glycolysis, sorry. So this one will go into the link reaction. Okay. Oh, wait one second. Link reaction. Again, I'm writing all these notes down, and I'm going to have this PowerPoint for you to download and keep as notes if you need it. It probably won't help, but might. Alright, oh yeah. Yeah, so fats definitely make more ATP or energy than proteins. It's just a given. The lipids are so long that they store more energy. Proteins don't do that much. Yeah. Alright, so let's do a recap of everything. There's a lot going on in this, you know. Alright, so first step, we have glycolysis. Glycolysis, if you remember, happens in the cytoplasm of the cell. And what happens is that you have the 9-step process, and that's you have glucose converted into pyruvate. Okay, so then you have stage 2, or the Krebs cycle, or the citric acid cycle. They really lump together the link reaction and the Krebs cycle, but I consider them 2 different steps. That's how I was taught. So, all right. So again, you have pyruvate that makes it through 2-ish reactions, it makes it into acetyl-CoA. This acetyl-CoA will go into this really complicated cycle and make all of this stuff: NADHs, ATPs, CO2s, stuff like that. Alright, and then you have the 3rd step. Again, through a series of steps like this, and then you have 2H2O. So oxygen is really the final electron acceptor. And so why we need oxygen is because without oxygen we would not have a final electron acceptor, and the electrons would have no place to go, backing up the without oxygen it will just back up all the process in the electron transport chain. Then it'll back up the Krebs cycle, and then that will back up the glycolysis. And then so you have a you have a high concentration of hydrogen ions here. Those ions will go through ATP synthase and make ATP. Alright, and as I've said, the electron transport chain, as you probably know, makes a whole lot of ATP compared to everything else. I mean, it's not even, like, measurable. It makes about 36, while everything else makes about 2 or 4. Alright, so what if we didn't have any oxygen? Well, I mean, we already talked about what would happen, and it would back itself up and stuff like that. But there are some circumstances where we don't have oxygen, but we still need to make ATP, right? Now let's talk about that. So what happens is that you have 2 types of reactions that can happen other than the ones, the 3 steps we've talked about. The 2 steps being alcoholic fermentation, and then you have lactic acid fermentation. So alcoholic fermentation happens in like yeast and other stuff like that. And so basically what happens is that you make alcohol. It regenerates NADH really. But we'll talk about how that happens and why. So what basically happens is that you have glycolysis happen. So you have, sorry, no, 1 second, let me just erase this. Alright. There. Alright, so you have glycolysis happen. And what happens in glycolysis is that you have every single step happen, and except for the step before pyruvate, when pyruvates are made, you have alcohol being formed. And so that alcohol will then be, the NADH, by doing that, you have NADH being made. That NADH will be sent back there, and then you'll have more ATP being made, and you'll just try to regenerate NADHs, and then send them to the electron transport chain. I mean, obviously you don't make as much, but it's better than nothing, right? And then lactic acid fermentation is what we as humans use. You know when your gym coach says, like, when your muscles are sore and stuff like that, your gym coach, you go up to your gym coach and he says, yeah, it's lactic acid buildup, we have to release the lactic acid. That means there's a buildup of lactic acid, and it'll make our muscles sore. And so what happens, what you have to do, what happens is that you have, again, in the same thing happens. You have, instead of making pyruvates, you make more NADHs and just try to recycle those NADHs as much as possible, and then send them to the electron transport chain. Alright, and so again, like I said, it regenerates NADH. Alright, thanks for watching. If you found any problems in what I said, or if you have anything to add, stuff like that, just let me know by telling me in the comments below. Alright, and also be sure to hit that subscribe button. It'll really help me out. And if you really like this video, share it with your friends. That's good too, And if you have any suggestions, let me know in the comments below. Again, if I made any mistakes, and that's definitely possible, just let me know in the comments below. Anyways, I'll see you guys in the next one.